Today we're going to talk about Prokaryotic Diversity, Chapter 4. When we consider the habitats and functions of microorganisms, we know that microbes are found in every environment on the planet. Also, we know that microbes are abundant on and in our human body, 10 to 1 compared to human cells. The functions of microorganisms include recycling elements. Remember, they're the great decomposers. Nitrogen fixation, bioremediation, and symbiosis. The living together, those symbiotic relationships. Let's talk about symbiotic relationships. Remember, symbiosis is when a species interacts with another one, or they live together, they have a relationship, and there are varying types of relationships that they can have. And these are the ones that we are going to learn in microbiology. The first one is mutualism, when both of the species benefit. We can think about a zebra, and sometimes you will see a zebra has these little birds riding around on its back. And what these little birds are doing, they're eating the bugs that are on the zebra. And so they're getting food from the zebra. And the zebra is benefiting because these little birds are removing these bugs that could be harmful to the zebra. So they both benefit. When we think about amensalism, when one species is harmed but the other is unaffected, we could think about penicillium or bread mold. When mold grows on your bread, like penicillium, it can destroy some bacteria. So it can kill the bacteria, harm the bacteria, but nothing happens to the mold. It's unaffected. So commensalism would be one species is benefited, but the other is unaffected. So a good example of that would be a monarch butterfly. A monarch butterfly can extract this toxin from a milkweed plant. And this toxin that it can excrete and store within itself is harmful to other vertebrates. It is a cardiac glycoside, and it doesn't taste good to other things like birds, and it can also cause problems for these other animals. So when it excretes this toxin from the milkweed, it protects itself, it's benefited, but nothing happens to the milkweed. It just has this toxin in it, and the monarch butterfly is just using what's there. It doesn't kill the plant, but it helps to protect the butterfly from things like birds. Parasitism is a relationship when one species benefits by harming the other. And an excellent example of this would be a bacteriophage or a virus. A virus, remember, has to have a host to survive. It has to have a host in order to replicate. It uses that host. Unfortunately, when it gets into the host cell and it uses the machinery of that host cell to replicate and it releases to the body, usually when the virus releases to the body, it destroys the host cell that it has been in. So the virus benefits by using this host cell to replicate and release to the body, but it destroys the host cell in the process. So it harms the host cell. So bacteriophages or viruses are called parasites. Now, neutralism, this relationship is when neither species is affected. They just live in the same ecosystem. Like you could think of like a scorpion living in the desert near a cactus. Neither one benefits from the other. Neither one is harmed by the other. They just live in the same area and just exist together. Here again are those two symbiotic relationships that we talked about on the previous slide. Mutualism, the little birds riding around on the zebra, both of them benefiting, one getting food and one being protected from whatever kind of bug is on there that might cause it harm. Commensalism is that relationship that can occur between the monarch butterfly and the milkweed. It extracts that toxin and protects itself from other predators, but nothing happens to the milkweed. It's okay. So, commensalism. One benefits and nothing happens to the other. Now let's talk about taxonomy and systematics. We use taxonomy and systematics to approach the diversity of organisms on this planet. We need to identify them, classify them, and name them. And that's what this is going to be all about. Let's talk about the principles of taxonomy: identification, classification, and nomenclature. And these are three very important concepts that you will need to know. The first one is identification. It's the process of characterizing the organisms. What is this species? Is it a dog? Is it a wolf? Is it a coyote? They all seem very similar, and we need to be able to identify them. Classification is creating order with these organisms. It's the process of arranging these organisms into groups. We can place them on the phylogenetic tree of life that has the domains of bacteria, archaea, and eukaryotes. We can place them on the tree of life based on their genetic makeup, how similar they are to each other, or how different they are to each other. Once they're on the tree of life, we're able to see how closely related is Salmonella to E. coli. How closely are they genetically compared? Once we can classify them and arrange these organisms into their groups, it can help us immensely. Nomenclature, if you remember, is the universal naming system that has the genus and the species. And we'll talk more about each of these individually. But remember, nomenclature just absolutely helps us to understand what something is by its name. It simplifies taxonomy. Why does taxonomy matter? This is something that you need to consider for future quizzes and exams. And the first ones we're going to talk about is how we need to identify pests. Pests that can occur in agriculture or pests that can occur in our forests and the harm that these types of pests can cause. We don't want an unknown pest destroying a crop of corn that we need for our food chain. We need to be able to protect our agriculture crops, and we need to be able to identify what might be harming them so that we are able to get rid of these pests. We must identify them. We also need to identify the pests in our forests that can be destroying the trees. The pine beetles have wiped out countless acres of trees in our forests, and that can contribute to wildfires. If you think about the gorge wildfire that had occurred, it was because it spread so rapidly because there were so many trees that had been destroyed by these pine beetles and the spruce budworm. There's all kinds of different pests that can destroy both our crops and agriculture and our forest resources, and we must be able to identify them. Here is an example of the destruction that a pest can cause to our forest resources. This photo was taken in the same general area during different years, and you can see the effect that has occurred by the pests that have completely destroyed and killed these trees that are in this same area, and it can increase the potential for wildfire. It spreads much more rapidly through dead trees than it would through a healthy natural forest. So we must identify this pest in order to get rid of it so more of our natural resources are not destroyed and we do not increase the threat of wildfires and the destruction of habitat for animals and bugs and birds. We need to be able to identify these pests to protect our resources. Another reason why taxonomy is so important is that it can save lives. According to the National Health Service in the UK, 1 in every 6 patients is misdiagnosed. Taxonomy can prevent the wrong diagnosis. Taxonomy can then prevent the wrong treatment. If you're misdiagnosed, you could be receiving a treatment that is actually harmful to you. Or you could be receiving a treatment that's not going to work because you've been misdiagnosed and you're not receiving the right treatment. I have seen misdiagnosis in multiple ways in my own life. My mother was misdiagnosed with something very minor and then ultimately died from breast cancer when I was very young. My son was misdiagnosed when he was an infant. And to this day, he has to deal with a lot of medications for epilepsy. And perhaps if he had been diagnosed earlier, this might not be happening to him. I was also misdiagnosed. I have an autoimmune disease, but I was misdiagnosed with other autoimmune diseases because they can look similar. But I underwent treatments that were very harmful to me, and I did not need those treatments because I had been misdiagnosed with a certain autoimmune disease. And ultimately, I didn't receive the right treatment and have developed more complications because I wasn't treated earlier. So we must have taxonomy so that we can save lives, so that we can diagnose accurately, so that we can provide treatment accurately, so that we can help people, how we can support patients in the right way. We have to have good taxonomy. Taxonomy makes us safe. We don't yet know what all is out there in existence. But during this time of increasing antibiotic resistance and the occurrence of viruses that are new and novel that we've never seen before, we need to use taxonomy, identification and classification of the things that we discover in order to see if we can use them for medications and vaccines. 95% of our medications come from natural sources, and so using taxonomy can help us build upon these medications that we develop and the vaccines that we can develop. We may not ever know what all is out there. Understanding how species are related can give us clues for how to control unidentified things. If we haven't identified something down to its specific species level, we can see how it compares to other things, how it's genetically related to something on the phylogenetic tree of life, how it may be similar or different to other organisms. We can use what we do know for identification and classification. A recent study showed that half of all Americans consume herbal supplements of some type. Now, herbal supplements can be very beneficial for some people. Some people may not want to take traditional medications, but want to try herbal supplements to see if those might help them for many different things. I even take some herbal supplements that I really think have been very beneficial. The problem with herbal medications are that they're not regulated in the same way as pharmaceutical drugs. So one company may produce an herbal supplement that has a completely different amount of the same herb compared to another company. Or it may produce a supplement that has very little of something in it. It is just not regulated and monitored the same way. So unless you know that the company has a very strong reputation, you may not actually know what you are taking and the amounts of what you are taking. Also, there have been many instances of incorrect taxonomic identification, and this can lead to increased side effects when you take herbal medications. You might be thinking that you're taking something, but if it has been misidentified and put into that supplement, you might be taking something that could be actually harmful to you instead of beneficial. So taxonomy is very, very important in the use of herbal supplements. Also, most people, when they go to the doctor, they do not disclose to the nurse, the medical assistant, the physician, they don't disclose the use of herbal supplements. And a lot of these herbal supplements can interact with regular medications, with a prescription you might be given. And if you hadn't talked about what herbal supplements you are taking with your medical provider, you might be causing more harm to yourself than good. Let's look at this photo of two different types of plants. Now you can see how important it is to get taxonomy just right. They look really similar, don't they? Now one is a carrot and one is not a carrot. One is poisonous. So let's look at that. Can you decide which one is the actual carrot and which one is not? The one on the left is the carrot. The one on the right is poison hemlock. So you could see they look pretty similar in some instances, and they could look really similar out in the wild. And if you were to take the poison hemlock and eat that instead of the carrot, then you could become very sick. So you can see how they could be misidentified. You may think that all giraffes are the same, but in actuality, there are different species of giraffes. They have different living requirements. They have different habitat requirements. They have different food requirements. They have different needs in order to be successful and to prevent their extinction. And we need to consider taxonomy for animals like giraffes and other animals on the planet for our future. We need to be able to identify and classify animals so that we can help them to be successful in their environments and successful in their existence. So what all is involved in a name? What does that mean? What is taxonomy? We're going to look at each of these individually, but let's start out with what they are. First, you need to know what it is. That's identification. What is this? What am I looking at? Then you need to know how it fits into the tree of life. That's classification. You classify what you are looking at. You group it. You put it on the tree of life within its classification. And you need to follow the nomenclature rules. Nomenclature is the name, but binomial nomenclature has a series of rules that you must follow. When we think about the identification of microorganisms, it is often done for reasons of health. We need to identify microorganisms to treat infectious disease. We need to prevent infectious disease. And we need to research new treatments. So taxonomy can really help us, especially identification. When we think about COVID-19, all of these are essential to help us to survive this pandemic. To help to control COVID-19, we need to be able to identify it, treat it, prevent it, and research new treatments and vaccines. It's absolutely essential. Identification of microorganisms should not be underestimated in their importance for human life and health. We need to be able to identify microorganisms in contaminated water. There are many developing countries who do not have clean water, and we need to be able to identify the microorganisms that could be making populations sick. We need to be able to identify microorganisms that could be in spoiled or contaminated food. We need to be able to identify them so that we can trace outbreaks back to where they initially started. We need to identify pathogens in clinical settings. We need to be able to use tests and techniques in order to identify what might be making someone sick. Let's look at classification now. So the goal of classification is to be able to place an organism, to group it according to its genetics on the phylogenetic tree of life. We need to determine their genetic similarity. We need to be able to look at their similarity, look at their differences. And when we're looking at their genetics, we're looking at their genotype. Now, you have to think that in the past, we had to only be able to use phenotype, what it looked like, how similar were they in the comparison of how they looked. But as technology has developed, we have been able to become really focused on the genetics of an organism and really identify how closely organisms are related. And as technology improves, as the science evolves, this will continue to be even better, and organisms will be placed on the tree, and we will have confidence in where they are and how similar they are to those organisms that are around it. We still have to consider the complication of horizontal gene transfer, though. Remember, I talked about how a bacteria has a pilus, and it can share its genetic components with another bacteria or with an archaea. It can trade these genetics, and because it sometimes can complicate classification because there can be a mixture of a bacteria or a mixture of species as well. All right, let's look at nomenclature again. Linnaeus, remember, he developed this standardized system of naming so long ago, but it still exists today. It is the binomial nomenclature, and it has these rules which you need to know. You must have the genus and the species. It has two parts to its name. You must capitalize genus, and species is lowercase. It usually sounds Latin, like it's a Latin word. It must be italicized, or if you're handwriting it on paper, you can underline it. Genus can be abbreviated, but species cannot. You need to learn these rules very clearly so that you can identify correct names on an exam. Phylogeny is the study of the evolutionary history of organisms. It is the evolutionary history of the relationship of species. Phylogenetic trees represent the number of genetic differences between species. Where they are placed on the branch of the phylogenetic tree is mathematically determined by these differences. Now, I talked about phenotypes before. That's how something looks. So they used to characterize organisms based on how they look because that's what their technology was at the time. That's all they could do. There was no technology to do genetic typing. There was no genotype, so they had to do phenotypes. But now we have that technology. And we can look at an organism's genotype and we can place them on the branch of the tree where they should go based on these genetic differences. Now, the goal was to do a species inventory of all species of life on Earth by 2025. I don't think that will be happening. It's a great goal to have, and it's something that scientists are working on, but I think it's going to take a lot longer to identify all the species of life on Earth. I've talked a lot about phylogenetic trees and classification. Let's really focus on that now. So think of the phylogenetic tree like a family tree. You might have a family tree. Think of it like that. How closely you are related on that family tree. So the line lengths or the branches are based on the genetic distances or differences between the organisms. So the longer the branch or the longer the line, there are more differences between those organisms. The farther away, the more differences they have. The majority of diversity is among single-celled organisms, which is amazing. The majority of the diversity is on microorganisms like bacteria. Now remember, on this phylogenetic tree system, there are many types of different domains. You could have the 3 domain, which is the bacteria, the archaea, and the eukaryotes. You could have a 4-domain tree that has bacteria, archaea, eukaryotes, and viruses. Or you could have a 2-domain tree. That was the one that just had the bacteria and the archaea. And all those eukaryotes were grouped into the archaea domain. So let's look at this phylogeny. Let's determine what species is the closest relative to an annelid. So up on the left-hand top part there, you can see the annelids. And we need to look at the branches. What branch is closest to the annelids? Is it the mollusks? A. Is it the roundworms? B. Rotifers? C. Or arthropods? D. The answer is arthropods. They are closest to the annelids on the tree. Those are the closest together on the branches. They are not similar to flatworms, even though they have a worm-like appearance. They are not similar to roundworms. They are actually close to arthropods. You have to look at the tree and you have to think that this is genetically based, not how they look, not phenotype anymore. It's based on genotype. So for this particular tree, the annelids are closest to D, arthropods. As I've said before, there is often a lot of debate in microbiology, and the domain debate will go on for a very long time. It still continues, it's still happening. The one that we focus on the most is the 3-domain system. That is the domains of bacteria, archaea, and eukaryotes. I like the 4-domain system because I study infectious disease. And remember, the 4-domain system has bacteria, archaea, eukaryotes, and viruses, because I really believe that we should be able to group viruses somewhere on a tree. There is also the 2-domain system. It's that Eocyte hypothesis, where eukaryotes are grouped within the archaea. So that's the one that just has the bacteria and the archaea. So here is how that tree would look with the 3-domain classification: the archaea, the bacteria, and the eukarya or eukaryotes. And you can see there are different branches within each domain. And so those closest branches would be the most similar organisms. This is the 4-domain system: bacteria, eukarya, archaea, and then the offshoot of the viruses in red. And again, they all have branches within the domain. This is that 2-domain system. You have archaea and bacteria, and the eukaryotes are grouped into archaea. Let's look at some names of bacteria and also consider those nomenclature rules. So remember, it has to have the genus and the species name. The genus is capitalized. The species is lowercase. It's usually sounding kind of Latin-y, and it must be in italics or underline. Those are very important rules that you need to know. Now, sometimes bacteria are named for the people who discover them, and sometimes they are named with descriptions that help you to know more about that bacteria. When we look at Klebsiella pneumoniae, we can see that Edwin Klebs discovered it, so it's named for him. If you look at Salmonella, Daniel Salmon discovered it, but he named it for himself. It wasn't based on that it looked like a fish or that it was salmon colored. It was because of his name. But if you look at something like Streptococcus pyogenes, it tells you a lot about that bacteria. From the first part, the genus, which is capitalized, Streptococcus, you can see that it is coccus bacteria in a chain form. Strepto, remember, means chain. Coccus would be the little round bacteria. So round bacteria in a chain is Streptococcus. That gives you a picture of what it looks like in your head. Pyogenes, which you'll come to know, pyo means pus. So this is a chain of round bacteria that probably causes pus when it infects someone. So sometimes when you look at the name of a bacteria, you'll know that it's named for somebody, or it actually will tell you something really cool about the bacteria. Let's compare and contrast 3 important species that we are going to consider in microbiology. Now remember, the eukaryotic species is a group of closely related organisms that have a true nucleus. They also have membrane-bound organelles. The prokaryotic species do not have a nucleus. They also do not have membrane-bound organelles. 2 definitions that you need to know are clone, which is a genetically identical copy of an organism. This will come up throughout the class. A strain is a genetic variation, a subtype of a particular organism. And we're also going to look at this a lot. Now, a viral species is a population of viruses with similar characteristics. That's kind of a vague definition, but something that you do need to know. So what is a species? A species is a specific and basic taxonomic unit. And as new species are discovered, there is often great debate, and scientists will often not agree on what that particular species is. But a species, by definition, is a group of organisms that can reproduce with one another and form fertile offspring. Species also have very common characteristics, a shared ancestry, and evolutionary history. So let's do a few review questions. Horizontal gene transfer makes blank tricky in microbes. A, identification. B, classification. C, nomenclature. The answer is B, classification, because horizontal gene transfer can occur through that pilus from a bacteria to a bacteria to an archaea, and they're sharing those genetic components. It makes classification tricky, because classification has to do with that genetic makeup of that organism, the genotype. Okay, ignore the extra is in this question. Is S. typhi an accurate scientific name using binomial nomenclature rules? Remember those rules. It does have a genus and a species. It can be abbreviated, that genus can be, yeah, that's true. But what is wrong with that? What is wrong is that it is not underlined or in italics. So this is not an accurate scientific name using binomial nomenclature rules. The answer is no, B. During the off-season, the flu virus infects birds and mutates slightly. This would be an example of A, how clones develop, B, how strains develop, C, how both clones and strains develop. Well, it can't be A because a clone is an identical copy. If the flu mutates slightly, which means it changes a little bit, that has to be B, how a strain develops. And a lot of times when we consider influenza, we have a wide variety of different strains. And we will talk about that in the third part of the class when we talk about infectious diseases. When we think about classification, again, it can be a little bit mixed up when two species interbreed. Like when a whale breeds with a dolphin and makes a wholphin, when a tiger breeds with a lion and makes a liger, or when you have a zony, which is a zebra mixed with a horse, and it's a zony. So there are hybrid organisms out there. People do breed different types of things sometimes, or sometimes they do breed in nature this way. But this can also kind of mix up classification. Not just horizontal gene transfer like with bacteria and other bacteria and archaea, but also species can intermingle sometimes. I want to bring to your attention some information about Eukaryotic Kingdoms because you might have heard these terms before in different biology classes. These are not terms that we use because we consider kingdoms to be outdated because they just aren't big enough, but I do want to cover them a little bit. So different eukaryotic kingdoms that you might have heard of include Animalia, Plantae, fungi, Protista. We look at these in different ways in microbiology, as you will see in upcoming chapters. But in general, Animalia are multicellular, they have no cell walls, and they're chemoheterotrophic organisms. Plantae are multicellular, and they have cellulose cell walls. They are usually photoautotrophic. Fungi can be unicellular or multicellular. Remember, fungi have chitin in their cell walls, and they can develop from spores or hyphal fragments. They are chemoheterotrophic. Protista, which is going to get confusing in later chapters, but I'll try and keep it reasonable for you and help you to understand it. Protista is a random group of eukaryotic organisms that don't fit into other kingdoms. It's like this group of organisms that they just stuck into its own little category. Protista. So these are eukaryotic kingdoms. And I wanted to talk a little bit about them because you might have heard them before and you're wondering, where are the kingdoms? We really focus on domains and subcategories in those domains. So that's why we haven't talked a lot about kingdoms. We focus more on the domains. The Burgess manuals have been the gold standard for classification of bacteria for many, many years. Initially, when the Burgess Manual of Determinative Bacteriology was developed, they identified bacteria based on their morphology, based on how they stained, gram-stained or acid-fast stains, on biochemical tests. They were able to look at things structurally. They did not have the technology to look at the genetics of these bacteria. But as technology has progressed, so has the Burgess Manual for Identification of Bacteria. And now they have the Burgess Manual of Systematic Bacteriology. And they use RNA sequencing in addition to all of these different tests to identify bacteria and archaea. And there is an extensive list of approved names of bacteria. And because it's so large, it's not kept in a book anymore. It's kept online. And all of these prokaryotes that have been discovered and named are placed on this list. And you can find all kinds of determining characteristics. Yes, their morphology, and yes, how they stain and how they are affected by biochemical tests. But really, they really focus on that genetic component now and use RNA sequencing in order to classify different types of bacteria. So remember, phenotype is how things look. How do they physically look? What are their characteristics when you look at them, when you stain them, when you do something with them? So the phenotypic characteristics that you can use for identification purposes of bacteria, you can use microscopy. You can do a gram stain, an acid-fast stain. You can do a capsule stain. You can do all kinds of different stains in order to look at the morphology and the arrangements of different types of bacteria. You can look at that morphology, also how it grows, the size, the color, the border. You need to look at different types of ways that bacteria can grow on an agar slant or on an agar plate, and you can identify its growth. You can use selective and differential media and see if it can grow in salt or if it can grow in blood. You need to be able to use different tests to see how this growth occurs and what it looks like. You can do biochemical tests. You can see how it metabolizes different things like sugars. So you use different tests to see how the pH changes when you do these tests on these different types of bacteria. So you can look at all kinds of different phenotypic characteristics of bacteria and identify them through these things. That's what we do in our microbiology class. Now we do these tests individually. We'll do a gram stain, and we'll do an acid-fast stain, and we'll do a capsule or an endospore. We will do sugar fermentation, and we'll look for hemolysis on blood. But now they have developed these commercial biochemical kits that can be used in hospitals for rapid identification of bacteria. Instead of doing each of the tests individually, they can use these fancy combined kits, and they can actually use robots to identify some types of bacteria with the use of these kits. For your purposes, when it comes to a Western blot, what you need to know is that we use gel electrophoresis to determine if a patient has a particular antibody present to a known bacteria. So using the patient's cellular fluid or blood plasma, we are able to see if agglutination occurs when it comes into contact with this known bacteria. It can be a very expensive test, and so it's usually done to confirm something. Someone, a physician, or a medical professional has already determined that they believe that a patient has something, and so they use a Western blot to confirm if the antibodies are present in that patient's serum. So the two key illnesses, infections that we are concerned with when it comes to a Western blot are HIV and Lyme disease. So if you are asked a question about a Western blot and what infections can it determine a patient has, for your purposes, you're going to say HIV and Lyme disease. And it's where we look for the antibodies to see if there is agglutination, which means clumping, when they are combined. So we're looking for antibodies and agglutination for HIV and Lyme disease. When we think about classification of organisms, remember we want to look at that genetic component. And there's many different things that we can do. We can look at the ratio of the DNA base composition between organisms and see the similarities and see the differences in these ratios. We can use what's called DNA fingerprinting. Often, DNA fingerprinting is used to determine the source of certain types of infections. It can be used to determine the source of a nosocomial hospital-acquired infection. It can be used to determine the source of maybe an outbreak of salmonella that was found on some type of produce or something like that. They can trace it back by comparing the genetic components of the bacteria that is found. They can use RNA sequencing for this genetic component. We look at the RNA components of a bacteria. PCR is polymerase chain reaction, and what it does is it amplifies the DNA. It makes more of it. It makes it into a larger quantity so that it can be used to do tests on it so that you can determine the genetics of the bacteria. Sometimes you need to make more of something, and polymerase chain reaction amplifies the DNA of whatever organism you are analyzing. Another way to look to see if organisms or species are similar or related, we can do nucleic acid hybridization. So if they are related, You could imagine that if you separated the DNA strands from the two species and put them together, that they would fit together. It will hybridize the DNA strands. So the greater the degree of hybridization, the greater the degree of relatedness. And the next slide really shows this very clearly. So let's look at that. When we are looking at the nucleic acid hybridization, we are looking at the genetic similarities in two separate organisms and their DNA. So we take the DNA from each organism and we heat it up, and it separates the strands of DNA. Then we take the DNA from one organism and match it with the DNA of another organism. If there is complete hybridization, if it matches completely, they fit together completely, the organisms are identical. If there is only partial hybridization, then the organisms are related, but they are not identical. If those two strands of DNA do not fit together at all, there is no hybridization. The organisms are unrelated. A southern blot is not like a western blot. The western blot was used to look for antibodies, and it was used for diagnosis for infections like HIV and Lyme disease. What southern blotting is a modified hybridization technique that is used to detect specific sequences of DNA. We are looking for certain types of DNA, and we are comparing them with the southern blot. A southern blot is a method used in molecular biology for the detection of a specific DNA sequence in two DNA samples. So southern blotting combines the transfer of electrophoresis separated DNA fragments to a filter membrane and the subsequent fragment detection by a probe for hybridization. They compare these two fragments, again, just like in the nucleic acid hybridization technique, to see if they match. That way they can tell if the two samples are the same. In this sample, they are looking to see if these two bacteria samples are Salmonella. So, southern blotting uses a probe and a gel material, which is different than the nucleic acid hybridization. What you need to know about southern blotting is that it is not like a western blot, which is used for diagnosis. It is specific to determining sequences of DNA and if they hybridize. Here are a few other tests that we can use for identification purposes. A western blot is a type of serology test. It uses the blood serum from a patient and examines the immune responses. It's looking for the antibodies. We can look at a patient's blood serum in different types of serology tests like a western blot. We can also look at a bacteriophage and a bacteria together. A bacteriophage is a virus. It is a predator of bacteria. So bacteria are susceptible to these bacteriophages, to these viruses, and we can examine them through phage typing. We can look at the cell walls and the plasma membranes of organisms through fatty acid profiles when we look for species-specific fatty acids. In flow cytometry, a bacteria is suspended in fluid, and we can measure the physical and chemical characteristics when that fluid is filtered out. Now let's shift topics a little bit and talk about Archaea. If you look at this timeline, you can see that it hasn't been that long ago that we started to study Archaea. Remember, the problem with Archaea is that they are extremophiles. They live in very extreme environments that can't be duplicated in a laboratory. So we have to study archaea where they exist, where they are living. And in those extreme environments, we can study them and we can research them. Archaea are really interesting, but we're only going to talk about them briefly in this class. Now remember I talked a little bit about methanogens, and archaea live in the digestive tract of cows. And you might have smelled a peculiar smell when you've driven to the beach through Tillamook. So because these archaea live inside the digestive tract of cows and they emit this methane gas, in order to help the environment through bioremediation, we have been able to capture this methane gas and use it to produce natural gas. If you look at the photo that's really colorful in the right-hand lower corner, you can see different colors within that picture. Those are different environments for different types of archaea. They can live in extreme heat, like these extremely hot areas in Yellowstone. And they produce a different color depending on the metabolism of their environment. So let's talk about those extremophiles again. The hyperthermophiles like to be in really hot environments. Acidophiles, again, like to live in acidic environments. pH 2 is optimum. Methanogens we talked about with the cows. Extreme halophiles, remember, are those that like to be in very high salt solutions. 25% salt or higher. Remember that archaea lack peptidoglycan in their cell walls. It is called pseudopeptidoglycan, which is false peptidoglycan. They just didn't come up with a better name. Their RNA sequences are unique from both bacteria and eukaryotic organisms. They are still considered prokaryotes, just like bacteria, because they lack a nucleus. Remember, methanogens can be produced not just for natural gas purposes, but they are also used in sewage treatment. What's also interesting about archaea is some research says that they are part of the normal human microbiome, that they can be found in the colon, the vagina, and in the mouth. But remember, for our purposes, archaea have never been implicated in disease. Maybe periodontal disease, but the research is still ongoing for that. There are some problems with regards to research of archaea. Remember, current pure culture techniques are inadequate for archaea research. We have a really difficult time studying archaea in the lab because they live in such extreme environments. We are better off studying them in those extreme environments. Also remember that lateral gene transfer is possible between bacteria and archaea. A bacteria can share its genetic components with an archaea through its pilus. And research is still being done to determine whether archaea can cause infections or not. So that's the end of chapter 4.